Good morning, everyone. See, this is for BSc course, session 32 for paper 8 biochemistry. In this session, I'm going to tell you what will be the metabolic fates of pyruvate. See, in the previous session, we have discussed metabolism, what are the different steps involved in glycolysis or EMP pathway. So let me just brief you regarding that. You know very well when glucose enters into glycolytic pathway, it gives you two molecules of pyruvate in two phases, preparatory phase and payoff phase. Now we have come further ahead. Now you have a pyruvate from glucose molecule. We will see what will be the fate of this pyruvate, or in other words, what happens to pyruvate next. Okay? Now, the pyruvate molecules obtained at the end of glycolysis enter into different pathways depending on the prevailing conditions. So, what can happen to this pyruvate? It can undergo alcoholic fermentation, it can be converted into lactic acid, or it can be converted into acetyl coenzyme A. And you will also learn about gluconeogenesis in this session. Okay? Now, we'll take one by one the fates of pyruvate in this session. Let me start with the alcoholic fermentation. See, this occurs in the absence of oxygen, so we call it as an anaerobic pathway. Okay? What happens in this? See, here pyruvate is broken down into ethanol and carbon dioxide. And this reaction takes place in two steps. In the first step, pyruvate is converted into acetaldehyde. See, this is your pyruvate, CH3CO, COO minus, is converted into acetaldehyde. What has happened here? See, this CO2 is removed. This CO2 is removed. So, it's a decarboxylation reaction catalyzed by the enzyme pyruvate decarboxylase. Catalyzed by the enzyme pyruvate decarboxylase. And this acetaldehyde is now converted into ethanol. Acetaldehyde is converted into ethanol. So you have an aldehyde group here which undergo reduction to this alcoholic group. In turn, NADH is reoxidized to NAD+. NADH is reoxidized to NAD+. So in this reaction, you have pyruvate converting into ethanol through two steps. Okay, so this is known as alcoholic fermentation. So look here, see this is a CO2 group which is eliminated by the enzyme pyruvate decarboxylate. So what we have got here is an aldehyde, and this acetaldehyde is converted into ethanol. Now aldehyde, upon reduction you end up with an alcohol. Okay, acetaldehyde undergoes reduction with the help of an enzyme alcohol dehydrogenase to give you ethanol. In turn, NADH gets reoxidized to NAD+, which is very essential. So, in yeast and some other microorganisms in anaerobic conditions, in what condition? Anaerobic condition, pyruvate is converted into ethanol and carbon dioxide. Remember, this process is known as alcoholic fermentation. As I explained you earlier, which takes place in two steps. In the first step, pyruvate is converted into acetaldehyde by the enzyme pyruvate decarboxylase, and acetaldehyde is converted into ethanol by alcohol dehydrogenase. Remember, this reaction reoxidizes NADH into what? NAD+, which is essential for the continuation of glycolysis. Now, we'll go for the next one. Now you know very well pyruvate is broken into ethanol and carbon dioxide, okay, which is an anaerobic pathway. See, the second fate of pyruvate, it can be converted into lactate. Pyruvate can be converted into lactate, okay. Now, this is also an anaerobic pathway, means which occurs in the absence of oxygen. So, it's a single step reaction which occurs in muscle cells. And in microorganisms, this is a normal reaction which occurs during the formation of curds. Okay. See, this is your pyruvate. CH3CO, COO minus is converted into lactate. This is converted into lactate. Okay. What is the enzyme used? Lactate dehydrogenase. See, from glycolysis, you have end up with pyruvate. This pyruvate is converted into lactate. See, this is a ketone group. CO group is a ketone group. Upon reduction, you end up with this secondary alcohol, CHOH group. And in turn, NADH gets oxidized to NAD+. So, this is also very important for the continuation of glycolysis. So, which is the enzyme employed here? Lactate dehydrogenase. So, the role of lactate dehydrogenase is to reoxidize NADH into NAD+. Okay. So, here pyruvate is converted into lactate. What is the importance of this reaction? So, this reaction produces ATP when during intense physical activity. When ATP cannot be produced by Krebs cycle, at that juncture, this reaction is very helpful. So, here regeneration of NAD plus takes place by utilizing NADH. See, the lactate converted here can be converted into pyruvate, okay, when oxygen is available. That is under aerobic condition. See, in some anaerobic microorganisms, say lactic acid bacteria, lactate is a normal product of glucose metabolism which takes place in the souring of milk. So, this is the one. So, this is pyruvate, CO group, ketone group, reduced to a secondary alcohol group. So, conversion of pyruvate into lactate. Okay. So, we have seen two fates of pyruvate now. One is broken down into ethanol and carbon dioxide. Second one is conversion into lactate. Which is the third one? This pyruvate form is converted into acetyl coenzyme A or conversion of pyruvate to acetyl CoA. See, the earlier two I mentioned were the anaerobic pathway, whereas this is an aerobic pathway, means which takes place in the presence of oxygen. Okay. And why this reaction is important here is this pyruvate which is obtained from glycolysis, the reaction should continue, isn't it? So when pyruvate is converted into acetyl CoA, this acetyl CoA enters into Krebs cycle or TCA cycle. So this conversion of pyruvate to acetyl CoA is a link between glycolysis and Krebs cycle. Okay. Now, how does this take place? This pyruvate is converted into acetyl CoA with the help of an enzyme complex, pyruvate dehydrogenase complex. So, this pyruvate dehydrogenase PDH enzyme complex has Mg2+, lipoic acid, TPP, NAD+, coenzyme A as cofactors, along with the enzyme pyruvate dehydrogenase. Okay. See, in this reaction, the two molecules of pyruvate obtained from glycolysis in the presence of oxygen are converted into acetyl CoA. Okay. So this reaction yields two molecules of carbon dioxide, 2 acetyl CoA and 6 ATP. Okay, from which is obtained from NADH, which enters into electron transport chain. So remember, conversion of acetyl CoA, that is, conversion of pyruvate into acetyl CoA, is the link between glycolysis and Krebs cycle. So you have seen now three things: conversion of pyruvate to ethanol and carbon dioxide, alcohol fermentation; pyruvate into lactate, lactic acid fermentation; conversion of pyruvate into acetyl CoA, and this is a very important reaction because it provides the link between glycolysis and Krebs cycle. See, eliminate the CO2 and at that place you have attachment of this acetyl CoA group. This is a coenzyme A group. So what you get is acetyl CoA. You have got is acetyl CoA from pyruvate. So they'll ask you for 2 marks. how is pyruvate converted into acetyl-CoA? So here you just cannot write the reactions in words. You, as a student of chemistry, you should know how to write the reaction. So pyruvate is CH3CO minus. Remove this COO minus. That place you attach SCOA. So what you got is acetyl coenzyme A. Okay. So now let me just briefly explain you what is this gluconeogenesis. Okay. See, this gluconeogenesis is the synthesis of new glucose. Synthesis of new glucose. So it's an aerobic pathway. It is an aerobic pathway and it takes place in the liver cells. It takes place in the cytoplasm of liver cells. Okay. Now, remember, gluconeogenesis is the synthesis of glucose from a non-carbohydrate source. You know very well, glucose is a carbohydrate. Here, carbohydrate is synthesized from a non-carbohydrate source that is known as gluconeogenesis. Now, see if you just look at these reactions, glycolysis, this is the overall net reaction. Glucose is converted into two molecules of pyruvate. Okay. Now, what is gluconeogenesis is two molecules of, pyruvate is a non-carbohydrate moiety. It's a non-carbohydrate compound. Non-carbohydrate is converted into glucose. Non-carbohydrate is converted into glucose. Here in glycolysis, glucose is converted into pyruvate. So, conversion of a non-carbohydrate moiety to a carbohydrate moiety is known as gluconeogenesis. Now, just now I told you this pyruvate can be converted into lactate, right? Pyruvate can be converted into lactate. Now, the cycle involving the synthesis of glucose in liver from skeletal muscle lactate. Under stress condition where this lactate is produced, this muscle lactate, this is a synthesis of glucose in liver from the skeletal muscle lactate, and in turn the reuse of glucose thus synthesized by the muscles for what? For energy purpose is known as Cori cycle. Okay. Now, see the importance of this reaction is, see any activity involving the muscle requires energy. Okay. And where does this comes from? ATP. Once it is converted from glycogen through the process of glycogenesis and glycolysis. See, when there is a lack of an adequate oxygen supply, okay, when usually typical as a result of any intense muscular activity such as running, when energy is released through anaerobic metabolism. So lactate produced, where it is produced from? Lactic acid fermentation. That will be absorbed by the liver. And there it is converted back to pyruvate. And in turn to glucose. Okay. So this glucose can then be used to replenish the levels of glycogen via glycogenesis and in turn provide ATP via glycolysis once the muscular activity has ceased. It is known as gluco. So this is what the overall reaction takes place in gluconeogenesis. Okay. So now, if I brief what all you have learnt in this session is, from glycolysis you have got pyruvate, two molecules. Pyruvate has come there. From pyruvate, what next? So from pyruvate can be converted into an alcohol, can be converted into a lactate, or it can be converted into acetyl-CoA. This acetyl-CoA formation links, or it is the link between glycolysis and the next one, that is citric acid cycle or the Krebs cycle. And you also know what is gluconeogenesis. Thank you.